It's Candidella imbricata as well, so we might be able to just confirm with a snap.
Oop. Sorry, video. Make it quick. Some dust coming.
Laser's clear.
Going in.
Oh, a small crab there. Yeah, so this is definitely another Candidella imbricata. Okay.
Cool.
Good species ID back here. We can.
All right, but you're probably not going to get a beauty shot out of that one.
But I have noticed that kind of on, if we follow this ledge directly backwards, there's a bunch of other fans on the left side of this little rock mound.
So follow up the far side?
Yeah, that's right. And I'm specifically, I have laser focus on the colony right at the top of the screen.
Copy that. Gives me a good direction.
That looks different as well.
It's my fault.
No, you're good.
Right there, Washley?
Yeah, that's right.
All right, video. You can start to come in.
Laser's clear.
It might just be a very sparsely branched Candidella. No, I'd say it looks like the same Candidella imbricata.
So
usually the colonies are pretty small and bushy. They don't get much bigger than that last one we just saw. That more fan shaped. This one is more sparsely branched, but polyp structure looks the same. Okay, thanks, pilot.
Copy that. If you want more from that video, just let me know.
Full screen.
Should probably get a ship move in his general direction.
Video's clear.
Yeah, I thought we had to move in.
We're gonna have to.
Oh, we do.
We're probably gonna have to see a lot of these over the course of the day today. They have.
How long was that? 20 meters?
They are probably the most abundant
30.
deep water coral in this area. At least by my counts.
Looks like current's
It's not
moving
the
up slope.
direction.
I think it's just lingering more than really blowing anywhere, unfortunately.
It's a good thing we're moving.
Yeah.
Get lasers back on.
Aye, aye, Captain.
Like
like
fish, upper right.
in the upper at 1 o'clock.
We don't have a lot of room to go that way.
Yeah, video go from
There's
here.
the branching.
That's about all we got right now.
We have a little room to sneak in if you want it, but not too much.
Looks like a halosar, yeah.
It's been a while since we saw one of those.
It's like a carnival game at this point. Get the lasers on the fish super far away.
Yeah.
All right, video, let's let him go. He's swimming away.
All right.
All right.
He's a little far off to the left-hand side in
Yeah.
the distance. There's something branching coming up, sticking out of the sediment.
You've got a floating holothorian here.
Where?
Behind you.
Oh.
Very helpful, I know.
Yeah, just going slow. Kind of went from a lot to this one thing. Pretty quick.
This is the most enigmatic scleractinium I've ever seen. It's just, it loves growing in sediment. You know, I always thought for a while when I was doing surveys of seamounts that maybe this is just some rubble that fell down slope and this has just happened where it grow and it's doing well, but actually this species of Madrepora it's almost always on soft bottom, growing on its own rubble.
Do you see, I mean, are they
video,
relatively large colonies? Because we've been seeing only small clumps of them.
Yeah, this is about as large as they get.
Okay.
Kill lasers or
You know, this framework is kind of very sparsely branching. You know, it looks like most of these polyps are alive, at least where it's white. The skeleton is white. But yeah, you can see the pinkish dot kind of in the center where the tissue is most dense.
But it doesn't seem advantageous for this because if you look at the skeleton, some of it is covered in sediment or debris. I mean,
yeah, definitely. It is odd. You know, they must have a pretty accelerated growth rate in terms of deep water corals to kind of last in this type of very difficult environment where everything's trying to attach and get up high up into the currents, and it probably wants to take over your colony. Okay. Can we get lasers on this entire cluster here just to get an estimate how big it is?
Yeah, copy that.
The squat lobster too.
Lasers
Couple
are
of squat
on.
lobsters. You know, I wonder, you know, since I usually see it on slopes and exactly like this, how much material is actually below the sediment. Because when we picked up that sponge the other day that had the mass, it was just a huge 10, 20 centimeters of rubble.
what we thought was that the coral was an associate of the sponge, but we found out that the sponge was an associate of the coral because it was growing on the dead skeleton.
Yeah.
Co-pilot.
I'm still perplexed by this because I was expecting.
Oh yeah, I was going to ask
not this
halfway to
sediment
bottom
at
if you
this
wanted
site.
some.
Yeah,
upper right.
we're still trying to acquire this slope. I think, you know, we are still actually kind of in the pit of this small valley or canyon. So we haven't actually made it that far, but
Like snaps in this
We're
guy
going
here.
to.
one day.
It's a good contrast.
It's an isopod.
It's kind of a teaser for what's coming later on today.
Yeah.
Follow me to the midwater.
Right, video. I'll stop there. We'll let him fly out. Nice. Come on. Yeah, copy that.
Here's one of the Medusa. Oh, shrimp. I thought it was a Medusa at one point.
Let's take a peek over here and then center up. Get going. Is that another shrimp video? Yeah, copy that.
Wow.
Oh, look at how cool. It's laying out its appendages to kind of float along.
Doesn't look like it's doing anything with its appendages. It's not feeding. It's just kind of cruising.
Close.
It's like drag.
I'll just let that.
Those are the
do its
longest
thing.
tentacles or antennae I've ever seen on a shrimp. They've got to be 20 centimeters.
Oh, fish.
He's going to look quick. We'll get the next one.
Something scooting fast across the bottom.
Oh, the dark.
Oh, it's a jelly
Oh,
floating.
here's a Medusa. A dark one. Kind of looks like an alien ship.
Yeah,
The right,
spaceship,
bottom
flying
right.
saucer. Well, maybe this is a good sign for the. Oh, there's
Yeah,
another
plankton,
Medusa, right?
Yeah. There are two types.
This might be a good sign for
Yeah.
our midwater team. Oh, both of them. Oh, they are the same. Or very similar.
There we go.
Copy that.
Very nice.
All right, Nav. Let's see, I'm sitting, 45 in Sirius.
Synchronized swimming.
Upslope still looks,
This would be a stock sponge.
yeah. We'll do another 30. We like, that's pretty good.
By the way, can we snap zoom on
Yeah, video you want to
right
snap?
at the lasers? Yeah. Looks like it has something orange on top.
I think that's there's like a little nubbin that some of these highly animated
Video's
sponges
still
have at the top.
approaching this from a distance. So if this gets more interesting, I'll be closer soon.
This looks very much like the one we sampled.
Yeah, I'm
Yeah.
still kind of far.
A
Just give me a
few.
sec.
It was much shallower though. But this more striking thing to me is that this sponge, despite how big it is, doesn't have any zoanthids on it, which they were encrusting on at least 99% of all the colonies we've seen.
And I
on
think
this
when we
expedition.
were in the beginning of this dive, I think I saw some stalks with zoanthids, but this one seems not to have any.
Yeah, just a couple of different
Some polychaetes.
yep, serpulid worms.
So it's the top.
It's really pretty.
Oh, sorry. I'll stop. Tilting down.
And that structure on the bottom looks interesting. The kind of the vertical stems on the sediment, on the sand,
Yeah,
in
copy
the
that.
background.
Yeah, we're just get a full shot real quick. We'll move to that next. All right, video. Zoom in and I'll center the shorter stocks.
I wonder if that's
I want to say, it's a urchin, I think, buried. Can it be?
does look like it has some color under there, but
It's brownish, or is that sargassum on top?
yeah, that might be Phytodetritus. I don't see any of these structures moving, so they look too uniform. They look possibly like some sort of infauna tube, maybe.
Some weird tube stuff.
Weird tube stuff. Worms do weird things.
That yellow tube, these tubes.
Yeah, I mean, really weird.
Yeah, they are small, like little
I'll
paddles.
zoom back on
It's
it.
interesting. There are like no openings at the ends, which is even a little bit more strange. You would think
Yeah.
there would be some type of opening if it was a worm.
We're getting a good look now in the foreground of these bundles of fibers that make up the sponge stock. They're called glass sponges for a reason. They do make fibers that are basically made out of a very pure form of silicon dioxide, or even the same material that glass is made out of.
All right, Washi, it's happy here?
Very much.
Yes,
All right.
thank you.
Video happy?
Let's lead with lasers.
Lasers on. And we're leaving.
It's 2 holothurians to the right, pink and purple.
Washi, any interest on holothurians?
Yeah, can we do a snap zoom in the middle? There, I believe there are 2.
Yeah, copy that. That video, you want to start to come in? Just focus on the bigger one.
Yeah, it looks like it could be the same species. One's just a little bit smaller.
Just float from this to the other one.
They have enough food here.
Lots of sargassum and phytodetritus and small organic sediment clusters.
Yeah, this one looks like it's... I thought it was going to take off.
A lot of the texture you're seeing on the
Video,
seafloor is
come wide.
from fallen marine snow, kind of gently accumulates on the seafloor. We were seeing some marine snow on our descent through the day today.
Copy that. Huh. I don't really see anything. Is this of interest at all, Watch-Its?
I think
I think it's the same.
yeah, we can skip that one.
Copy that.
It looks
Moving
like
on.
it does have some zoanthids on the stalk.
Go switch. We're in for like 30 minutes of no bottom. Switch real quick. It's a good time. Watch-Its, we're just going to do a quick pilot shift.
Sure.
Okay. No. So we're slowly reaching the ridge that we should be climbing pretty soon. Actually, can we snap zoom on this sponge right in the to the right of the lasers? It has different type of.
Yeah. Can snap in to see if it's something we want to take a longer look at. Thanks.
Okay, yeah, that's okay. It seems to be the same. It's just we're getting a
Okay.
top view.
Great. Ooh, there's a fish. Yeah.
Ah, there's some fish.
Let me see if I can get it in view. It's up pretty high. It's up out of the lights.
There's also another fish right at like 9 o'clock.
Yeah, I see it. I'll go over there.
There's something also big. Looks like maybe a sponge.
Yeah. Copy that. Mosey that way. Lots of little holothurians spread around. You want to start in partial before it darts off.
Okay.
Video.
I think
Ah,
this
looks
is
like
the
a
one
halo
that's
star.
chill.
They're
Okay.
pretty common.
I'll sneak up on it. Let this zoom is good. Scan it with the lasers once. Then we can kill them. Looks good.
And can we, when you're finished with the fish, just go back down to look at the sponge? There's a branching
It looks pretty scarred up.
organism right to the right of the sponge.
Yeah, copy. We'll come back to the sponge in a little bit.
Thanks.
Just get a close look at this since it's swimming off.
Ready for more zoom?
Yep. Just matching it.
Up.
Full wide.
He's attacking the vehicle. Yeah, he's coming into view.
That would make the fish people very happy.
Yeah. Well, a little dust on the foot as I backed up to get back to the sponge, but
We want to come into it on the right where the coral is.
copy that. Just trying to get away from the dust cloud coming down. Scan it with lasers once. Looks good. Thanks, video. Yeah, I'm gonna nudge a toe here. Come on in.
So we have a couple things here. We have a glass, the larger base type sponge, looks like a glass sponge. And then there's an anemone to the bottom left. And also another type of glass sponge above the anemone that's a yellowish color. We've been seeing these in perforated sponges. They're in all different colors, kind of purplish to opaque to yellow.
You can take it if you have it.
Yeah, so you can see the mouth part of the anemone. This is a corallimorph?
Kick on the lights.
I think it's a corallimorph. The knobby structures on the end of the tentacles are kind of just jam-packed with nematocysts, and they deliver a pretty potent sting. Usually you find corallimorphs with these knobby structures, not so much with anemones, but I always wondered what there's a very significant difference between corallimorphs and
to the
anemones.
coral to the right.
Walking to the right.
Internally, anatomically, but just from looking at them on the outside, there are some characteristics that we can usually identify with corallimorphs, like the knobs.
Copy. It's a little obstructed from whatever that is. But
Yeah, so this is another primnoid octocoral. Actually, there's a little worm or something down at the base. Can't tell. It looks like maybe a shrimp. This is most definitely Candidella imbricata.
okay.
Yeah, thanks for that. It's a weird sponge too.
Yeah. There's, it looks like 3 types of sponges possibly,
Oh, yeah.
or at least 2.
Yeah, the Farreid-like sponge, that yellow and translucent.
Let's go with that little squat
Sometimes
lobster on
purplish.
the top
Sometimes
left of the
purplish.
sponge.
Yeah, I'm not sure if that is the same species or even in the same genus. That's a pretty phenomenal
Squat lobster.
depth
I see
distribution range.
I think this
think
is the
it's
first time
it's
we've
a
seen them
brittle star.
Little
yellowish.
star, yeah.
Oh, actually.
Probably key.
These guys.
Ah, look at
Just frame it.
Amphipods and
Frame 'em
isopods.
both.
Ophiuroids. And a big window in the side of the sponge. Probably just a growth, something in its growth. Although, we did see sea stars for dating on sponges. I wonder if this might just be like a, it seems like a very big spot, too big for a sea star or an urchin to eat.
Those amphipods don't seem to be moving. Could this sponge be eating them?
It's, it's not something we see very often. There is a polychaete in the lower left too. I just say it now. The glass sponges, the carnivorous sponges, do belong to the same group here, the hexactinellid sponges, but this is more likely a filter feeding sponge. But that being said, the glass spicules that we're seeing, they could have some sort of microstructure that makes it easy for the crustaceans to attach to.
Yeah, a lot of times they make these like sediment channels that they hide in.
Yeah.
So maybe the spicules help to keep them in place.
Pilot's clear. I see a big branching something off to my port. I'm just gonna. You could just kill the lights, thanks. Something
Oh, there's
over
something.
here. I don't know if it's
There
of
we go.
interest. I'll keep going. If you want to see it, we'll linger.
Yeah, we do want to look more specifically at the lower right coral colony here.
The smaller yellow?
Small. Yeah.
Okay. Interesting ledge looking. Something in the background there. I don't know. Not sure. Coming down. Got the lasers on it. Okay, video, start it.
The size of that skeletal material
Yeah,
is
the
very
right-
fascinating.
At one point I thought it was maybe a branch, but definitely looks like it's...
Yeah, so this looks like a plexaurid octocoral. The tentacles are retracted, the polyps are retracted. There's one asteroschematid, it looks like, ophiuroid. If I had to guess, I would say this is probably in the genus Paramaricia.
Full screen?
Did you see that?
Paramaricia is a good guess.
Brittle star looks like it was gnawing on the polyp there.
However, there is some similarity in some of the species of Paramaricia with Placogorgia, so I wouldn't be
the
able
lower
to
portion.
tell for sure
Let's
without
go to the base, see what's going on there.
without a better
Doesn't look like it's attached to this
There's
stick.
that other
view
Yeah.
or sample.
paddle
Just
stuff
hanging out on this stick.
to the base.
Oh, to the lower left.
Yeah.
Yeah. Okay. Did we get enough time on your full
Good
specimen?
observation video.
We did.
Yeah, definitely. I think what we have here is probably a part of a stony coral skeleton. Some of that material is in the sediment, and that these two corals have anchored onto that old coral skeleton and continue to grow.
Who's living on the bottom of this
I mean,
thing?
given how black and manganese covered this,
Slightly different looking.
it's a spiky crab. Can we
Is that
zoom in the
max
crab?
zoom?
Is Max
We got any more?
Get any closer?
I can get a little closer. See if I can.
It's a crab I've seen before, but it hasn't been identified yet. It was collected in 2014, and we affectionately called it spiky crab
Spiky crab.
for lack of a better term. But it's not a squat lobster. It's get a little bit closer and confirm before I tell you the story.
Okay, come in. See if I can find it again. There it is. It's very small.
Lower lights.
Yeah, it's
Slide it
very
low.
well armored.
Standby video. At the edge of my tilt, I got a little too close, backing up a little bit. Okay.
So if this is the same crab, pull up a.
Just backing up a smidge. There we go. Try to stabilize again.
Yeah, so I can't tell. It's pretty
Any more
small,
zoom?
but it's not a
Yes.
squall lobster.
Take it.
It's probably a
Next
lithodid.
zoom.
Okay. Try to
The
stabilize.
lithodid crabs are known to be extremely spiny. And I wish I could find that photo of the adult.
It's very small.
Yeah, thanks, pilot. I think that'll suffice.
Okay.
See, maybe we can find a bigger one. But yeah, it was a lithodid crab, possibly in the genus Neolithodes, that we had sampled that looked very similar to that. Much larger though, in 2014. And from what I understood, it was possibly a new species at the time, so I haven't checked in on that lately, but it could be worth a second look after this.
Picking up and moving on.
There it is.
Yeah, it's much more spiky.
Yeah, as an adult, they're very spiny.
You want to come port? Okay. Sounds good. Just gonna push through this sediment. Those lights are out, right? Thanks. Yeah, I'm happy with it. Scan this with the lasers, it looks a little bigger. Yeah, I don't think we're gonna linger. I was just looking at size, looks
Yeah, when
similar
they're
to ones we've
young,
seen.
I noticed that they're very white, and then when they're a little bit older, they turn
Caramel colored.
caramel, yellowish.
I think I see some
Is that
possibly
rocky,
hard bottom
rocky
over
hard
here.
bottom over there?
Yeah. I'm gonna.
Little tilt up and show it to you more.
It's all right. Let me just get a little closer here.
I think it's a crest, and then past
Gonna-
the crest.
I think it comes
goes
down a little ways
Yeah.
the hill, so I'm gonna come around.
Something dark on the upper right, like a fish shape.
Okay. It's out of reach at the moment, but hopefully it lingers long enough for us to take a look. Just around. Get the ship moving a little bit closer to this, so I can have a glance, and as the ship moves, make some progress on it.
White Olithurian at the bottom.
Yeah, I see that. Come down
Yeah,
a
there's.
little bit.
What's your sense of the current in this area?
Most of what you're seeing is me driving pretty quickly right
Okay.
now. There's not a lot of current that we've observed so far. Yeah. Thanks.
This rock type looks actually very different from what we were seeing just a little bit lower.
Yeah.
This is like a fresh
I don't
looking
have it.
rock.
I don't have quite enough tether to get around the other side of this ledge yet.
In geological terms,
Yeah.
you always have to make that statement.
There's a yellow fin in the foreground that might be of higher priority if you can get there. Or is that
A
a
yellow
yellow
fan?
sponge?
I think
Oh,
it's
the
a yellow
sponge, yeah.
sponge.
Ah, okay.
Yeah.
That's all right. Can look at the other things though.
looks like there's some failure because there's a little bit of rock rubble in the channels.
Yep.
I'll mosey along slowly as you guys do.
Ah, it looks like the black coral.
Oh yeah, small
Yeah,
piece.
small
definitely.
piece. We just collected, maybe.
Yeah, it has that branch coming out of the kind of the top.
Keep an eye. I'm going to see if I
Third
can
of
make
it.
it up to this possible fish.
So this is something different because you were noticing that at the other site in Mona Canyon that most of the corals were
Okay.
attached to the rocks that were coated
Yeah.
by manganese. It's
Yeah.
so hard to say. But these, now you're seeing them on and there's not as much, but there's still some corals attached to this fresh coral.
Yeah,
Yeah.
yeah. There is like
looks
a
a little
wall
bit
here,
more
but it seems like kind of
consolidated
the wrong direction for us.
generally than we were seeing at
Yeah.
Mona Canyon, which could mean that relative
Yeah.
to Mona,
You want to
which
come in on it?
seemed to be. Oh, there's a fish.
Yeah, it's just floating out there. I think
Ah.
I have some room to push that way if we want to see a closer look.
There's a parasite on the tail. I can see it this far away. It's huge.
Kill the lasers for now. Thanks.
Maybe a parasite on the mouth too, in the lower right. Or is that
I think that's
scar?
food.
Food or a scar? Yeah.
Should be getting closer.
Maybe an isopod on the tail portion.
Ah, there's another one on the other side.
Coming around where I have a little more tether so I can push out
Seems
to
to
it.
be one on each side.
I think
The
given
dorsal.
given the size of the first few rays on the dorsal fin, I think this might be a grenadier or a rat tail, but I'm not going to stick to that.
Okay. Oops,
I
sorry,
think
video.
we might be.
Coming up a little higher.
I would say this is
tilting
probably
down.
a new observation. I
As he
don't
gets
recall
closer,
this
I
one
have to pick up
No.
and let it
over the
go
past
down
few
on
weeks.
us a little. Stand by video. We'll get a good shot of it in a second. Just sneak up on it.
Has really big eyes. Usually they don't. They're not that big, and
It's very chill.
Okay.
The lasers indicate it's probably
Okay.
40 centimeters.
He's turned for
30 to
us.
40
I'm gonna
centimeters.
line him up.
Round numbers.
And
Now
we
we
can come in a little closer.
go back and try and measure that more accurate.
Copy.
I probably want you to stop on the white stuff.
Kind of limited on the
Tilt
without. Okay, I'll take a little tilt then.
That's a good
Almost
good
touching
shot
the
of
bottom.
a few amphipods attached to the fish, the dorsal ray, the fish, and a really nasty looking scar there. You can see there's a bunch of scales that have been worn away,
Yeah,
maybe
I'm
teeth.
gonna let him
full screen.
or some sort of
Come to the
large
screen a little.
parasite maybe used to be attached to this critter. Yeah, so this looks like a grenadier
Slight tilt down.
or macrurid
tilting down.
fish.
Get the tail. There, I'll hold that for a while.
And I think they're in they're gadiforms, right? No,
And
I
video's
think
clear.
they're gadiforms, not ophidiforms.
Okay. All set, video?
Yeah.
Video's clear.
All right, we're all set then.
Yeah,
Okay,
gadiforms,
move on.
the same group that cods belong to, codfish.
Looks okay
We
from
haven't
the
seen too many
fisheye,
of those,
but
actually. They're
No.
usually pretty common at these depths. We've seen a lot more ophidiforms and
all right, I have room to switch out
Allosaurs,
to
yeah,
that
they've been
hard bottom real quick. So
the
I'm gonna
most
go take
common
a
ones
glance.
on these dives, the allosaurs.
Bright white
Okay.
object on the lower
Rock
bottom.
seems to have changed
See
pretty
that.
rapidly here. Kind of now into this scoured carbonate-looking light color sediment and rock.
Oh yeah, it's a guy in a burrow, or something's cleaned off the surface there.
Oh, what is that?
Slowing down. Got a video, come on in. It is a little
Oh.
burrow.
Looks like a shrimp.
Shrimp in a burrow.
Shrimp in a burrow. Wow, it's a really carved out burrow.
Very
Yeah.
thorough.
I don't think it was a shrimp.
Yeah, I
A
mean,
He-Man shrimp.
I want to know whoever his landscaper is. These,
Copy.
I find it hard to believe that, you know, echinoderms are known bioeroders, but shrimps, I don't think they've ever been identified as being very strong bioeroders. I know they do make burrows sometimes, but this is just so fresh, and we
I'm
see
gonna
a lot
tilt
of these
down a little.
depressions and possible bioerosions
this.
or dissolution features in the rock, but
They're definitely made by animals, but I don't know if the shrimp would have made
go
this
full
unless-
screen.
yeah, I agree.
Look at his antennas in a minute.
Sometimes siderid urchins are bioeroders, but we haven't seen any in this area. Interesting. Well, he just got lucky then.
It's a good spot.
Moved into the neighborhood and there was an opening.
Probably get a good deal.
He has no neighbors. She.
Video's clear.
Pilot's clear. We will. Okay. All right. Take
We'll have
a
to see
quick
if we
glance
see
at
any
this. See if we got any more goodies on this face, and then I have to get back on track for the uphill. Okay. What are we heading? What were we heading? 110. Okay. Coming around.
Is that another hole?
I think so. There's something brown right here, and then it looks like another hole right next to the lasers.
We could take a snap on the hole, see if it's gives us an idea of what made it. Looks empty.
Some kind of burrow. Perfectly circular.
Yeah.
They're pretty deep.
Thanks, video.
Either the rock's not as hard as we think, or there's like a tiny little drilling team that makes these holes.
Oh, I should have nudged it a little with the toe. I could have told you how hard it was. I still can. Do you want to know how hard it is?
No, it's all right.
Okay.
I think we'll see some more.
All right.
It looks pretty hard, but we'll wait till see the next outcrop up here.
Copy. Yeah. You can hear the ship thrusters making a lot of effort. That will
So
affect the tracking.
while we have a little bit of
Extra
time, we're going to do
vibrations and
kind of our
turbulence and
introductions
stuff
that
like
we missed when we first hit the bottom today because there was just a profusion of corals. So in the back row as your co-science leads, I'm Steve Oskovich, sitting in the Watch Lead 1 seat. I'm a PhD candidate at Temple University
Copy 110.
in Philadelphia in the Department of Biology. I'm a specialist on the ecology of deep sea corals and a little bit about the biogeography of corals in this area. Oh, there's a fan there?
Yeah.
Yeah.
Sneaking up on it. Might be another
Careful, you might scare it.
bottle brush or something. Come in partial.
Uh-huh. Can we take a
Laser's
closer
clear.
look at this? If
Yeah.
if you have time?
Yeah, we've got time. Come down. Trying to get a little window to that bulge there. I see a little break in the branches. It
I think
might-
we're on the backside of an enemy.
Ah, okay. That makes
It's wrapped
sense.
around the branch.
Yeah, okay, I agree with that. Come on in. Just rocking. I'll settle out.
Oh no, that's an enemy.
Yeah. So what we're looking at here,
Some bamboo.
first I thought it was something else, but since it's growing in sediment, it kind of helps narrow down the possibilities. But what we're looking at here is a bamboo coral, and you can actually see where it's
Oh, yeah.
branching right on the nodes. This bamboo coral is pretty well known at being able to tolerate and adapt to living in a sedimented environment, but this is most likely Acanella, which is a bamboo coral that's known to be modified for soft sediment habitats. I'm not curious if we, there was one anemone and 2 squat lobsters here,
And a
Good to
couple
see.
shrimp.
1 squat lobster,
Yeah, there's a
couple
shrimp
big
up
shrimp,
and a
top.
couple
yeah.
large shrimp, yeah. At the base of these, it's kind of interesting. We'll be able to see sometimes these Acanella species, I can't tell at the moment, but they, some of the species have modified roots. Rather than attaching to the rock, they actually grow this brambly-like root structure that helps them anchor into the sediment.
I don't think that the sediment is very deep here.
Yeah.
This is all pretty
Yeah, pretty hard underneath.
hard underneath, yeah.
Yep. This is interesting.
Video's clear.
Okay, alright, that's clear.
Yeah, we can move on.
That sponge. Yeah. And a different type of Holothurian, it looks like.
Holothurian with a crown, I think.
Yeah.
You want to partial as you go over them?
Sure.
Ah, this looks up another Holothurian. Sea cucumber above and stalked glass sponge with a brittle star.
Okay. Continuing on.
What is this? Yeah.
Got a little
This looks
urchin.
very-
A bigger urchin.
Looks like it's on its side or something.
Come around and take a quick look.
Take a snap to see which side has got the feeding
I think
side.
it's
You
eating
can kill the
something.
lasers if you want.
We've had yesterday,
Come
if
on
you
in.
were tuning
Video?
in with us, we saw
The
a lot
feeding
of
side's on the
urchin
opposite.
predation.
Yeah, I was going to do a little circle for you here.
And this looks like another. This is a carnivorous sponge,
Yeah,
and it's looks like it's eating a carnivorous sponge. This is the first.
we've seen some wacky, echinoderm action going on here the past few days. I didn't think anything ate these glass sponges, the carnivorous glass sponges, but I guess they are now, or they have been.
This looks like a different type of siderid. I don't remember the test being so red in the others. I'm not sure if it's just a color variation.
Yeah, I think this is a deeper water species as well. Now, I was wondering if there looks to be a brittle star at the end of the sponge as well. I wonder if, since we were seeing a lot of echinoderms eating echinoderms yesterday, if they're actually also interested in eating the brittle stars.
I think brittle stars are pretty quick, though.
Yeah.
They probably detach one arm and move on.
Yeah, depending on how many arms they have wrapped up in the colony that they're holding on to.
Take a little more zoom if you want.
Maxi?
Yeah, this one looks like it's just eating its way along.
So far we've been seeing these sea urchins eat black corals, other echinoderms, and now glass sponges.
A note, one of our scientists ashore, I'll have to pull up the email, mentioned that this mysterious red spines is actually, we noticed, or we know from other stuff that our scientists ashore have told us, that these siderids are unique in that they don't have tissue covering the spines. Actually, look at this. There are some small serrations on the spine here, but you can kind of see them here and here.
Yeah.
But they're definitely not as distinct as Histosideris. But they don't have tissue covering their spines. It's one of the reasons why we find them so fouled. But those red spines, actually there is a small layer of epithelium that covers the spine until it's presumably full grown, and then it actually starts to slough away or fall away or retract.
That's clear.
So that's why we don't see the red spines with
anything
attached organisms.
encrusting over it.
I learned something new. I think I've learned more about sea urchins than I have about corals on this entire expedition.
They're definitely voracious predators that we've seen these past couple of dives.
Ready to get another short move in? Want to probably reach the star here.
Hey, pilot, I changed my mind actually about these corals in the sediment here up on the upper 12 o'clock position. There's another coral kind of sticking out of the sediment. I may want to go for a collection on that colony, since...
Sorry, I see a few here.
So
the larger one with the sea star on it, the black one.
Yeah, and especially because it's being eaten by a sea star.
Okay.
If we can get some imagery of that first.
Yeah, of course.
We'll take a look at the sea star, see if it's something.
Probably won't get around to the backside. Oh, you are. Great. Okay. Great. No, I think we're gonna sample this so you can hang tight. Yeah.
Oh, there's a sea pen too in the lower left.
Oh yeah.
Okay, video, come on in. Thanks. See if I can come around to this.
It's all closed.
This edge. Get as much of the feeding as I can. backing, sorry. Pushing ahead. Bring it back in view. There it is. This looks like a pretty good vantage. I'll land. Actually, I'm pretty far away still. You want me to sneak in a little closer video?
Yeah, it looks like it's averting its stomach here.
Yeah.
Okay, that looks close enough. On the brakes. Stabilizing.
We know this predation is kind of a slow process here. I was wondering if...
Cool. Great, thanks.
It'd be nice to know if this is a star that we've seen before predating on corals, or it's common to predate on corals. Very spiny goniasterid. Well, let's see. Stand by, pilot. We're going to consult the shore side here and see if it's a good idea to disturb the sea star to get the coral, because there may be other corals we can sample.
Slight tilt up.
Tilting up.
Meant for full screen.
Oh, I see. Thought you were going for the top. All right, I'll hold that. I don't think it'll fall off. Starboard inner is fine. It's pretty much like glued on there with its stomach. Roland, I can maybe get you in a few more degrees around.
All right,
It might
pilot,
be obscured by the coral a little bit, but
Video's clear.
this is watch lead.
go ahead, watch lead.
We're going to actually pass on this collection
Okay.
and see if we see another coral. We weren't able to get a good ID on the star, so we
Okay.
will see if we can find another one without.
Okay. Sounds good. We'll continue on.
We missed the sea pen though. I'll throw it over to you.
Okay.
Your turn.
I totally forgot.
We've been doing our introductions here in a pretty stepwise manner because we've been seeing so many interesting, cool things on the bottom today.
Okay.